Hello, my name is Chapman, and in this video, I will discuss my model of protein synthesis. For this model, I decided to start with the location of the titles and area of key information. This area of key information is at the top right area of the model that serves as a legend. In this, it depicts general details such as the creator's name, dimensions, and demonstrates which colors represent which nucleotide, such as thymine being represented with the color of red, as a form of key. The second step of creating the model is identifying and collecting the materials needed and also determining what parts of the process they'll represent. For this, I use a diverse range of materials. These include colored matches, yellow clay, string, pipe cleaners, foam paper, glue, and white cardboard as a template. For this, the color matches I used were the colors to represent the nucleotides of both deoxyribonucleic and ribonucleic acid. These include adenine, blue, uracil, purple, cytosine, green, guanine, yellow, and thymine, red. I used clay as a component of the model as it is a material that is easily malleable and can solidify easily. For the model, clay was used to represent the two processes of protein synthesis, transcription, and translation. I used clay to represent the RNA polymerase, ribosome, and tRNA. String in the model represents the shape of the strands of DNA and RNA, as a line or double helix. Pipe cleaner, specifically purple colored, was used to represent the final product of protein synthesis by being scrunched into a ball, and this represents protein. Foam paper was used to represent the amino acid chain, hence why there were numerous in the model. The final part of the model was glue, which is used to keep everything together the clay was not able to, even after solidifying. And as a template, the white cardboard was used to hold everything on it and to represent the information. After placing all the pieces, I then labeled what they were and outlined the process that occurred with each part of the process. For this model, I will also outline and guide through the steps of protein synthesis as depicted by this model. The first process that occurs in protein synthesis is transcription, which is within the nucleus of a cell. In this, an enzyme called RNA polymerase attaches itself to a strand of DNA. While attached, the RNA polymerase creates a strand of mRNA that is made of bases in the DNA. On a side note, different nucleotides pair with different nucleotides, such as adenine, blue pairing with uracil when the mRNA is created. This mRNA consists of a message made of RNA that has been based on the DNA. When this process is completed, the mRNA then leaves the nucleus of a cell and then enters into the cytoplasm of the same cell. In this area, a ribosome is then attached to it because it is attracted. Ribosomes are responsible for the creation of proteins. Also in the cytoplasm, tRNA molecules are present, in which carry amino acids and are able to bind multiple together. The tRNA brings in the amino acid and reads the complementary codons, triplet bases of the mRNA, and compares these to the anticodon, the triplets of the tRNA. With each codon of the mRNA, red, a new amino acid is created, with the type depending on the bases in the codons, such as methionine needing the bases of adenine, uracil, and guanine. With numerous amino acids, a polypeptide bond is created, which is a chain of amino acids. When the last amino acid is added, the chain then folds into a 3D shape to form the protein. This is the process of protein synthesis represented by this model. In conclusion, I've used a variety of numerous materials for the production of the model of protein synthesis, in which I've outlined all the necessary steps of this process.